Is biotechnology principle and processes. So now biotechnology, see the name itself, like something which is unique, like because you are in 12th standard and it's something like new for you, though it is not new, but you feel little bit new, that because there are certain how we can change the character of any particular organisms. So in this chapter, basically when we say biotechnology, it basically a kind of, we are using the techniques, okay, biotechnology. So it is a techniques of living, using living microorganisms, plant or animal cell, or we can say their either their component or enzyme for the organisms to produce the product and process that is going to useful for the human beings, and that is what the biotechnology. This is just a basic concept I'm talking about. Okay, what if someone asks me what is biotechnology, so I can simply say biotechnology means we are going to use some kind of microorganisms. So that microorganisms can be microscopic, or it can be a plant, or it can be animal cell, to change their manipulations to produce certain kind of product, and that product can be useful for human beings. That is what biotechnology, right? But let me tell you, this term biotechnology is not a new term. It was quite old terms, which is used by, or we can say this biotechnology term was coined by, this term was, what happened now? So this biotechnology term coined in the year of 1917, too much old, okay? So by the Hungarian engineer, the name is Karl Ireki. Karl Ireki, who first time used this term biotechnology. Okay, so I got response from moderation team that is student having exams, and that's why the number is very less. Okay, fine, no problem. So yes, now here, very first time this biotechnology term coined by Karl Ireki in the year of 1971, and later on, and he actually described, why he described the biotechnology, because he described a process for large scale production of pigs. Okay, so basically he was an engineer, so he worked on large scale production of pig, large scale production of pig. He used this term biotechnology, and then later on there are several what we can say scientists who come forward in the era of biotechnology and who discovered different terms. Like I can say gene manipulation. Basically when we're talking about the biotechnology, it is what it is, nothing but it is a gene manipulation in the past emerging sciences. Means because we have already knowing that sciences, but how we can manipulate them, that is what the basic idea behind this. So it is started with the development of recombinant DNA technology, and it is named various method. Like some say DNA manipulation technology, some say recombinant DNA technology, some say it's genetic engineering. So it have a different terms, but the most common term is biotechnology. And this is possible because there are two important discoveries based on bacteria, and that two important discovery the first one, that is called the plasmids in bacteria. Plasmids in bacteria. And the another one, that is called the endonuclease enzyme, restrictions endonuclease enzymes. Okay, these two discovery made possible biotechnology in hand to hand by the modern era. Okay, if this two discovery was not there, then definitely biotechnology is not so flourished as what we see nowadays. So these two discoveries very important for biotechnological processes, that is plasmids in bacteria and the restrictions endonuclease. RE stand for, it's a restrictions endonuclease. Okay, these two discoveries very important. Okay, now so we will discuss more about because our topic is biotechnology principle and process, so we will just learn that what is biotechnology, and we will also try to learn then what is the actual process of and how we can manipulate the entire processes that we are going to learn in this chapter. Now this is just to recall that what we studied in the last session. Last session means what we studied from the last chapter. So from the last chapter we have already studied about that how microbes in human welfare, useful in human welfare, especially in the field of biofertilizer, and in case of biocontrol agent, and in case of biogas productions. So this is common, and these are the different type of microorganisms which are used, like some are bacteria as a biofertilizer, some bacteria can be used as a biofertilizer, cyanobacteria and mycorrhiza. So if I say bacteria, so there are some symbiotic bacteria like symbiotic bacteria, some are isospirillum. These are the free living bacteria. They are associated, and they can form a symbiotic relation with the organisms. Another one, you can see the cyanobacteria, which include the heterocyst present. So without that, heterocyst has a capability to trap the nitrogen from the atmosphere. So that cyanobacteria, then anabina and azole. In case of mycorrhiza, we studied about the ectomycorrhiza and endomycorrhiza, especially glomus, right? We also studied in the microbes in biological control, biocontrol agent, which include the ladybird, who control the aphids, dragonfly, who control the mosquito, then Bacillus thuringiensis, who control the butterfly, caterpillar, then there is a trichoderma species, who control the plant pathogens, buffalo virus. So this all we have already studied in the last session. Yes? Yes or no? Okay. Now come to our topic, that is today's topic, especially we can say biotechnology. Now biotechnology is simple terminologies which we can compare with the old biotechnology and modern biotechnology. Now how we can say that old biotechnology is also referred as a traditional biotechnology. And we have already studied in the previous chapter that microbes, how we can use that microbes to convert milk into curds, right? So microorganisms were first used to produce some organic compound like citric acid. Even they are also used to produce the antibiotics. The level of production of penicillium yield has been improved, but have not been changed, and they remain as same as those obtained from the natural cell line. So in all these processes, only the natural capabilities of organisms and the cells are exploited, and these activities are called the old biotechnology. This is old biotechnology. Why? Because there is no any manipulation, because they are continuously exploiting the microorganisms, right, for production of biological product. So this is what the traditional method. Now what is the modern method in biotechnology? In the modern method, what we are going to do, we are going to use a certain kind of process through which we can manipulate the gene level so that we can get ample amount of product within a short period of time. Because older technology is time consuming, and it consumes lot of biomass. but in modern biotechnology, there is no such kind of what we can say processes. So therefore, the human we can take one best example like human insulin is also produced from the transgenic Escherichia coli strain that contain and express the insulin gene. Even the protein produced by the transgenes are called the recombinant proteins, and the production technology based on the genetic engineering that are termed as a modern biotechnology. And it started this technology, modern technology, that is in the year of 1970. Means before technology, and now after 1970 we can say this is the modern biotechnology. Now how you will define the biotechnology? So there are various definitions made by EFB, but the most prominent, most common definitions of biotechnology is that who cover both traditional view and modern. That is what called the actual definitions. Means if you want to define, your definition should contain the traditional point of view also and the modern point of view also. If both these techniques, traditional and modern point view, are compiled, that is what the actual definition of biotechnology. And it was given by the European Federation of Biotechnology. Now what is this European Federation? So here you can see biotechnology is the technique of using living organisms or their enzymes to product and process useful for human beings. That means of you can use one simple microorganisms for their product, and that product is beneficial for human beings. That is what the simple term of biotechnology. Now these biotechnology terms, you can see this is called the European Federation of Biotechnology, who define biotechnology as a integration of natural science and organisms, cells, parts thereof, and the molecular analogues for the product and their services are referred as a biotechnology. Clear? So the most prominent definition is that is the integration of natural sciences and organisms, cells, parts and thereof, and molecular analogues for the product and their services is referred as a biotechnology. Now these biotechnological terms, we can say, follow two important common factors. Okay, the first, that is use of biological agent, and the second, that is the product or the service is generated for well-being of humans. Okay, so it basically follow two important common factors. It is based on two common factors. The one, what I said, the one common factor, that is called the use of biological agent. Use of biological agent. Now what is mean of biological agent? Like any kind of microorganisms, if you're using, that is called the biological agent. Yes or no? So that is your biological agent. Another one, what is the product? So that product or the service is generated from well-being of human beings. Okay, so this is the two important terms. Product obtained from the biological agent. Okay, now biotechnology is not a new terms, as I told you, because it is from the ancient terms, right? So here you can see biotechnology is deal with the microbes mediated process. Now microbes mediated process means making curd, bread and wine. It is not a new technique. Making curd is not a new technology, right? It is the ancient technology, making curd, bread, even wine. Even this biotechnology is also deal with the in vitro fertilizations. What is mean of in vitro fertilizations? All the test tube babies you might have heard about, right? Because we already studied the reproduction in organisms, living I mean so human beings. So there is a test tube baby. Test tube baby is what? It is nothing but it is an in vitro fertilizations. And in vitro fertilization is a part of biotechnology only, because we are using techniques to create a better human beings, right? So that is in vitro fertilization, synthesis and using of gene, like any gene, if you are interested, you can synthesize and you can use that gene to incorporate with another organisms. So it is also dealing with the synthesis and use of gene. Preparation of DNA vaccines. Nowadays, you might have seen about the COVID-19 vaccine. So, lot of vaccines are prepared from the DNA only. So, it also deals with the DNA vaccines. Correcting a defective gene. Suppose any in human body or any organisms, any gene is defective. So, that defective gene can easily be removed by this biotechnological process. So, these are the five major category where we can see the biotechnology, how the biotechnology can deal with all these products. So first is microbes mediated process, in vitro fertilizations, synthesis and using of gene, preparation of DNA vaccines, and then correcting a defective gene. So these are the major areas of biotechnology where biotechnology can be utilized for the better for human beings. Now, as I told you, this biotechnology is based on two important techniques. That means of entire principle of biotechnology is based on two important concepts. One is called the genetic engineering and the bioprocessing engineering. These are the two important, we can say, the pillar of biotechnology. Without this, biotechnology cannot be possible. So genetic engineering and bioprocessing are the two core techniques that have been led to the development of biotechnology. Now, what is there in the genetic engineering? Let's try to understand. Now, here you can see, when I say genetic engineering, that means of it is a technique in which genetic materials is chemically altered and introduced into the host organisms. Now, what is mean of that? Suppose this is a kind of DNA which is extracted from the other organisms, and this DNA incorporated with the host organ. This is your vectors incorporated with the vectors, and then this vector is transferred into the host organisms. So it will multiply. That this is what the genetic engineering techniques. Here. Yes. Another one you can see, the chemical engineering. Now, what is chemical engineering or we can say the bioprocess engineering? So here, maintenance of sterile ambience, because that organism, the host is, it cannot be survive in the open atmosphere, because in the open atmosphere there are ample amount of microbes who are going to kill that host organisms, and therefore we need to maintain them at a sterile conditions. We need to provide certain sterile conditions, so that ambience in the chemical engineering process for growing the desired microbes or eukaryotic cell in the large quantity for the manufacture of antibiotics, vaccines, and enzymes. This is what bioprocess engineering. That means of, like suppose I can say I want to grow penicillium notatum. You know that what is the role of penicillium notatum? Penicillium notatum is a microorganism, a fungi, right? What it produces? It produces the antibiotics. What is the name of that antibiotics? Penicillium, right? Now, I cannot grow the penicillium in the open tray or any open place where I can supply the media. Is it possible? No, because in the open atmosphere there except the penicillium notatum, there are ample amount of microorganisms are present. So it is impossible to grow only the penicillium notatum in that open atmosphere, and that's why we need to provide them a sterile conditions where only penicillium notatum can grow. No other organisms can hamper the penicillium notatum. Are you getting my point? Yes. So what happened? This penicillium notatum, when you provide all the ambience atmosphere like a sterile area and a good chemical a good nutrient than even good at airy conditions than your penicillium product quantity as well as quality is also good and high. So that is come under the bioprocess engineering, that how you will use the technique to produce a large amount of penicillium instead of a small amount. So that technique is called the bioprocessing. So first part is you are going to do the genetic manipulation, and after manipulating you are going to provide them a desired area so that they can able to grow and give you the better product for human beings. Is this clear? This is the two important concept related to the principle of biotechnology. Yes? Any doubt in this? Okay. Now let's move with the next one. When I say the processes, there is, and that all possible, this genetic engineering is possible because there is a presence of plasmids in bacteria and the restrictions endonuclease. These are the two important things which was discovered at that time, and because of these two discovery, biotechnology is possible. So what that two discovery? The very first one, as I told you, that is plasmids as a vector, the first one, and the second one is that is the restrictions endonuclease enzymes, RE, restrictions endonuclease. These restrictions endonuclease enzymes going to help, I mean to say it is going to, these two discoveries, make a good role of biotechnology. And the very interesting things is that restriction nucleases is discovered by Arbor, Nathan and Smith. Okay, this restrictions endonuclease, let me write here, it is a restriction nuclease. It was discovered by Arbor, then Nathan yes. So it is Nathan and another one that is Smith, who discovered in 1970, and for this discovery they got Nobel Prize. For the discovery they got Nobel Prize on this in the year of 1978 in the field of chemistry. So in 1978 they got a Nobel Prize for this discovery, that is what called the restrictions endonuclease. Clear? Okay. Now here there is also one scientist which is generally remembered because of his technique to introduce the virus into the bacteria. Okay, and the name is that the Paul Berg, another scientist, that is called the Paul Berg. Paul Berg is also remembered because he developed a technique to introduce a gene of SV40 virus, SV40 virus into bacteria. Okay, because of this he is also called the father of genetic engineering. I mean to say the Paul Berger is called the father of genetic engineering. Yes, father of genetic engineering. Clear? And even he awarded Nobel Prize. He also got a Nobel Prize on his discovery. In 1980, he received a Nobel prize for his discovery that how the gene of SV40 virus can introduce into the bacteria. So these techniques, when he developed, he got a Nobel Prize. He also referred as the father of genetic engineering. Is this clear? Okay. So what is recombinant DNA? The process of joining and inserting the foreign piece of DNA into the host organisms to produce a new genetic combination is called recombinant DNA technology. And in short, it is also referred as RDNA technology. Okay, that is recombinant DNA technology. Now what is recombinant? So you can see here, Jason, please on your front camera. Camera off is not allowed. Yes. Sasidaran. Okay. Now here you can see what is recombinant DNA technology. This is the plasmid DNA. Okay. And in this plasmid DNA, suppose your gene of interest is this one. Yeah, Jason, what happened to you? Is there any problem? Jason. Jason, please tell me, is there any problem?
Sir, I asked my parents what about it, sir.
Okay, so you are using a cell phone.
Sir, no, sir.
Please keep your cell phone aside.
I didn't use, sir.
Okay. And please make a habit of making notes because that will help you. Yes? Good, good, good. Okay. So what I said that how actually recombinant DNA is created. So here you can see this is a plasmid and your interest of gene is this one. So you can use, now you have to cut this part, this segment, because this is your gene of interest. How you will cut? With the help of restrictions endonuclease enzymes. So this is kind of molecular seizures who going to cut these portions, and when you cut and this gene is going to introduce into the vector, then this is called the recombinant DNA, because this is the vector DNA, or we can say the cloning vectors DNA, and this is your plasmid DNA, which is your desired DNA. So that segment is going to introduce, and this now become the combination of DNA of both the gene, and that's why it is called the recombinant DNA. Is this clear? Or short, it is called the rDNA. Now let's move ahead. Now this recombinant DNA was produced by the Cohen and Boer. Cohen and Boer, who first time produced this recombinant DNA. Stanley Cohen and the Herbert Boer in the year of 1972. Right? Now the piece, or we can say they cut the piece of DNA from plasmid carrying antibiotic resistant gene in the bacterium Salmonella typhimurium, and this cutting of piece of DNA from plasmid was with the help of restrictions enzymes. It is also called the chemical scalpels. Okay, sometimes This restriction enzyme is also called the chemical scalpel, or either you can call it a molecular seizure, which is very commonly used, right? Now, the piece of foreign DNA cut from the plasmid was linked with the plasmid DNA acting as a vector. So, during the piece of foreign DNA with the vector was done with the help of enzymes, what that enzyme required to join that cut segment of DNA, that is called the DNA ligase. DNA ligase, right? So it is called the molecular glue. It is a molecular glue who stick both the DNA, the desired DNA and the cloning vector DNA. So it acts as a molecular glue. Now here you can see they isolated who isolated these two men, that is Cohen and Boer, who isolated the DNA, isolated an antibiotic resistant gene, a piece of DNA from the plasmids of Salmonella typhimuriae, a kind of bacteria, and it was linked with the plasmids vector and transferred into the E. coli. And what is the result? The gene was expressed and multiplied in the E. coli. So that means of those Salmonella, gene is expressed and it is created the multiple copy in the Salmonella. Clear? Now, what are the process? What is the basic steps in the gene cloning? So any gene of interest, I can say in human beings or in plant or any type of organisms, the first things of gene cloning is identification of DNA with the desirable gene. Means suppose you have to identify which one is the desired character of a particular organism. Like as I said, Penicillium notatum. So all the gene of Penicillium notatum is not going to produce the penicillium. So I'm interested in only those genes who have capability to produce the penicillin, right? So that is my desired gene. I identified as a this is my desired gene and I want to manipulate this desired gene. So here you can see this is the human cell. You have to just isolate the DNA from that human cell. Okay, this is the DNA and you have to what? You have to do the isolations. How you will do the isolation? That technique we will learn in the upcoming session, that how to isolate the DNA of any living organisms. Like even you can isolate your DNA by your own at your home also, right? So there are certain techniques through which we can able to isolate our DNA in laboratory, as well as if the basic things is available at home, then that we can also isolate at home level. So first thing is you have to identify your gene of interest. Okay, and that gene of interest you need to transfer in the desired vector, because sometimes vector should be suitable. Otherwise what happens, vector can have a right to reject that DNA, right? So first is identification of DNA with the desirable gene, right? Okay, now another you can see, so traditional hybridization technique lead to the inclusions and multiplication of undesirable gene and desirable gene. Like suppose those gene which is not desirable and those which is desirable, so you can multiply their numbers according to their requirement and not requirement. And in the genetic engineering only desirable genes are introduced, because my interest is for desirable genes. So what is the role of undesirable gene? Nothing, right? For me it is nothing, because my interest is only for desirable gene, which is my interest of gene. So always keep in mind, especially in the genetic engineering, you have to always introduce only those genes which are desirable. Undesirable gene is not required, because that undesirable what we can say gene is not will give you more productivity what you are expecting, right? And that's why this desirable gene should always be introduced into the genetic engineering. After identifying your desired gene, what is the next part? Now the next is introduction of the identified DNA into the host cell. Now you have to find out which host is more suitable. Like suppose this is your this is your bacterial DNA, this is a plasmids, right? So you once you created once you identified your desirable gene and introduced into the vectors, now this vector you need to introduce into the suitable host. Okay, and what is that suitable host? So you can see it can be a E. coli can act as a suitable host. So the vector DNA such as plasmids is used to deliver an alien piece of DNA into the host organisms. So this vector, why I need to introduce my desired gene into the vector? Because this vector have capability to introduce themselves into the desired host organisms, and that's why I have to first this plasmid introduced into the host E. coli, and this host E. coli later transformed and convert their multiple cell copies. So this is what the another step, that is introduction of the identified DNA into the host organisms. Now after and then what happened? This transformed cell produce n number of copies of your desired gene, right? Now third one is maintenance of the introduced DNA in the host and transfer of the DNA to its progeny. That is also a very challenging task for the biotechnologists, that how they can maintain the entire DNA throughout the sessions. So throughout their experiment process. So what are that process? So simply you can see, you just read out this diagram. Okay, this diagram represents, this is a human cell and this is the bacterial cell. So now in the human cell you isolated your desired gene, and from the bacterial cell you isolated the plasmid. Okay, now this plasmid you need to use certain enzymes to cut the plasmid at certain locations. So you can just cut it these portions, and then you have to attach this desired gene into this plasmid vector, and these plasmid vectors now it joined with the help of DNA ligase, and again you transferred into the host, and this is your bacterial cell for containing the gene for human growth hormones, and this bacteria will produce n number of gene that having capacity to for the production of growth hormones. This is what at the genetic level. So this is just a basic steps, means what are the actual process of cloning. Are you getting my point? Yes? Okay. Now, so there are total 3 steps in a gene cloning. Now, what kind of biological tools are required? Like if you want to perform these 3 process, what I explained, you need a certain instrument to carry out this experiment or a kind of project or a kind of we can say project executions. What that 3 types of tools we required. The very first tools, that is called enzymes. Without enzymes means what? Certain enzymes which are helpful to cut and paste the DNA, your desired DNA. Okay, for cutting you need some special enzymes. So you need a certain enzymes who act as a cut and paste mechanisms. Another one, you need a cloning vectors, that is called in simply we can say the vehicle DNA. Without cloning vector, it is impossible to transfer your desired gene into the bacterial host, and therefore you require a cloning vehicle or vectors. And the last one is competent host is also required. So today we will cover only the enzymes, and cloning vectors and competent host we will cover in the next session, because enzymes is so big. Along with the enzymes we also need to understand some gel electrophoresis, which is also used in this experiment or in this field of biotechnology to identify the certain characters. And that's why we will focus only enzymes in the upcoming and remaining two cloning vectors and competent hosts that we will cover in the next sections. Now let's see what is the enzymes and how it works. So you can just observe this diagram. What this diagram represents? This diagram represents, this is the DNA segment and this is a scissors who cutting that DNA segment, right? That means of you require restriction endonuclease, a kind of enzymes who going to cut the DNA into two equal parts or into the several segments, right? Now you also require a vector that is called PBR322 or a kind of plasmid vectors. Okay, then you also required, okay, so these are the important one is enzymes and then cloning vectors. Now let's move ahead. Enzymes. When I say enzymes, there are various type of enzymes used in this recombinant DNA technology. But the major one I'm listing here, the very first one is a lysine enzymes. Okay, lysine enzymes. Now what is this lysine enzymes? These enzymes are used to open up the cell to get the DNA from the experiment material. Like suppose this is your desired bacterial cell, right? And now you want to isolate the DNA from this bacterial cell. This is the DNA which you want to isolate. How you will isolate? You need a lysine. Lysine is what? It is a kind of enzymes who going to degrade the cell wall. Okay, so it destroy the cell wall. So if it destroy the cell wall, then what happen? This cell wall will remain disappear like this, and then you can use easily isolate the DNA from this bacteria. So this is DNA which is isolated, right? So for this enzyme is basically used to open the cell to get the DNA from the experiment. Even sometimes this lysozymes is usually used to dissolve the bacterial cell wall. It is always, keep in mind, lysine is always used to dissolve. I'm just writing here, lysine is used to dissolve the bacterial cell wall. Lysis, lysine means to degrade the cell wall. Clear? Yes. So that means of no any enzymes have capability as like lysine who can degrade the bacterial wall. So therefore, why you need to degrade the bacterial wall? Because you need to isolate the DNA. Yes or no? So to isolate the DNA, you need to dissolve the bacterial cell wall, and once it dissolves, then only it is very simple for you to extract the DNA. So you have to keep in mind that lysine is important. Now in the plant cell wall, they are basically made up of cellulose, while in fungi it is made up of chitins, right? So cellulase and chitins, for that you need to use a different enzymes, right? Cellulase and chitin you need to different enzymes. Like suppose I want to use, this is the bacteria, now I want to use a fungi. Okay, if you want to use fungi DNA, then which enzymes you are going to use? Tell me. If you want to use fungi DNA, how you will extract the fungi DNA? What enzymes you require to extract the fungi DNA? Chitinase. Very good. Why chitinase? Because fungal cell wall is made up of from chitin. Yes, absolutely. So fungal cell wall is made up of from chitin, and to degrade the chitin, we require chitinase. So chitinase is going to degrade the chitin wall, I mean to say a fungal cell wall, so that you can easily extract the DNA from the fungi. And if it is plant cell, then if you want to extract the DNA from the plant cell, then what you will do? Which enzymes you are going to use? Yes, Sindhuja Sri, Sarjana, Sasidaran responded. Okay, tell me, plant cell is made up of from, yes, plant cell is made up of from, okay, cellulase, 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 oh my god, cellulase. Is it cellulase or cellulose? Cellulose. Plant cell wall is made up of from cellulose, right? Yes, it is made up of from cellulose, and to degrade the cellulose you need a cellulase enzymes who going to degrade the cellulose, right? Okay, so this is lysine enzymes specially used for bacterial cell wall, to degrade the bacterial cell wall. Now the next enzyme, that is called the cleaving enzymes. Now what is cleaving enzymes? So cleaving enzymes are used to break the DNA molecule. Okay, cleaving enzymes are used to DNA molecule to break the DNA molecules. Now see the first steps. What is the first steps? I used lysine to isolate the bacterial DNA. Now this bacterial DNA is a thread-like structure, right? Now I isolated the bacterial DNA, but this bacterial DNA is a thread-like structure like this. Now I want to cut this thread-like structure. I can use to cut the DNA. I use a cleaving enzymes. This is what called cleaving enzymes. So with the help of cleaving enzymes, you are going to cut the a full a long fragment of DNA into a smaller fragment like this. This is a small segment of DNA. So how you will cut? With the help of enzymes. What is the name of that enzyme? It is called cleaving enzymes. Now these cleaving enzymes generally used to break the DNA molecules, but they are classified into three types. They are classified into three types. Endonuclease, and one more I forgot to write, that is called restrictions endonuclease. Clear? So total three type of cleaving enzymes are there: exonuclease, endonuclease, and the restrictions endonuclease. Okay, so these three enzymes going to cut the DNA into a segment. Now when I say exonuclease, they remove the nucleotide from the terminal end. So exonuclease, like suppose this is the DNA strand like this. Okay, this is the DNA strand. So where exonucleus work? Exonucleus is always work on their terminal end. Okay, so terminal end of the DNA in one strand of duplex. Like either it can be work on 5' to 3' or 5' to 5'. Means it always work on extreme position, and that's why it is called exonuclease. Are you getting my point? Like here you can see this is an exonuclease. Where it is working at the extreme position. It is not going to cut in the center portions. Yes, is this clear? Kirtini, are you there? Okay. Okay, another one. Please on your front camera. Kirtini, if you are there, then please on your front camera. What happened? Any problem? No? Okay, all right. So, exonuclease. Now come to the endonuclease. So, endonuclease, they make a cut at a specific position. That means of it is always going to cut at the specific positions. What is the specific positions? Like suppose this is a full band of DNA. Now I want to cut at a specific location. Okay, this is the band of DNA and I want to cut at specific positions, like at this portion. I want to cut from here. So this is my specific point. So for that purpose, I will use endonuclease. I will not use exonuclease. Okay, so always keep in mind, endonuclease is a very specific in nature. And because of this specificity, it is going to work on the specific location only. Okay, so these enzymes do not cleave the end and involve only one strand of DNA duplex. Clear? Okay. And the third one, as I said.
Sir.
Yes.
So what do you mean by cleave at the only one end of the duplex, sir?
Okay. See, what I said that in case of exonuclease, they remove nucleotide from the terminal end. Now, terminal end means what? Suppose here is and this end, right? So it is this exonuclease work on terminal end. Terminal end of what? Terminal end of DNA in one strand of duplex. Duplex means what? What is mean of duplex? What do you know? The basic characteristic. DNA is a double stranded or single stranded. Yes. Tell me your answer. Jason, right? Jason responded. Jason, are you there? Double stranded. Okay. So double stranded is a duplex. The another term. Instead of using double stranded, I say duplex. Clear? So, yes. So it is double stranded. Don't get confused yourself. Duplex means that exonuclease or endonuclease, they are going to cut at the specific location. Suppose it is endonuclease, it is going to cut at a specific location within the DNA. So these enzymes do not cleave the end, and end means it is not going to cut at the extreme position. and it involves only one strand of DNA duplex. So it is going to quite only one DNA strand, not all the DNA strand, because it is a very specific. Clear? Jason, I hope your doubt is clear, Jason. Okay, good. Now the next one, as I said, the restrictions into nuclease, right? Now here you can see restrictions into nuclease enzymes is also referred as a molecular seizures, right? So they cut the DNA duplex at a specific point, and their single strand DNA stranded free end are called the sticky end. Okay, that means of this is a DNA strand. Okay, it is going to cut at specific positions like this way. Okay, so this end is called the sticky end. Okay, so their single stranded free end are called the sticky end, which can be joined end to end by the DNA ligase. Means suppose if you want to join with another DNA, so it going to join like this. Okay, so that is the purpose of restrictions into nucleus, because it going to create a sticky end. That is the only difference in restrictions into nucleus, endonucleus and the exonucleus. Hope these three cleaving enzyme is clear to all of you. Yes? Okay. Now I will talk more about the restrictions into nucleus, because this is very important. Exonucleus, endonucleus is very common because, yes, Sasidharan, what happened? Are you getting bored? No? Then why you are moving back?
No, sir. I just called my mother to give me some water.
Oh,
I'm feeling thirsty, sir.
okay. So you can yes, take it, no problem. Okay. Now let's move with the next one. As I told you that I will talk more about the restrictions into nuclease, because the initial two enzymes are very common and it is very easy to understand, right? But restrictions into nucleases are something which is very interesting. Why? Because these restrictions into nuclease enzymes, which is called the molecular seizures or scalpel, and this was first time isolated by W. Arbor in the year of 1962 in the bacteria, and they called the molecular seizures or biological seizures. In 1978, Arbor, Smith, and Nathan, I think so. We have already, uh, I already told you about this. Three men who got Nobel Prize on their discovery of restrictions into nuclease. Right? Yes or no? So that is important. Now another one, that they recognize the base sequence at palindrome site in the DNA duplex and cut its strand. Means basically they found at the palindrome site of the DNA duplex. And you know very well what is a palindromic sequence, because they've already studied, right? So it is similar to, we can say it is similar to Escherichia coli R1. Now, what is this Escherichia coli R1? So, restrictions endonuclease Escherichia coli R1 is found in the colon bacteria of Escherichia coli, and they recognize this particular enzymes. The base sequence is GAA-TTC. So you can see here GAA. In this diagram you can see GAA-TTC. Right? They recognize this base sequence, okay, in the DNA duplex, and they cut its strand between the G and A, as you can see here. So you can see they cut from G and A. So this is the G and this is A, between G and A. See the red marker. Can you be able to see the red marker? It is in between the G and A. Cutting is made between the G and A, here and here, right? Now this cutting, so basically only restrictions enzyme type 2 are used in the gene manipulation for two regions. Okay, so there are different types like restrictions into nucleus, as I told you that this is the enzymes, but there are different types like type 1, type 2, type 3, like that. Okay, so restriction type 1, type 2, 3, 4 and 5. But out of this all 5, 2 are used. 2 are very important. Why these 2 are very important? Because they only restrictions enzymes type 2 are used in the gene manipulation behind gene manipulations, right? Why? Why there are several types? As I told you, there are several types. There are total 3 types. 3 or 5 types are there nowadays. Because initially it was 3, but now it is total 5 in number. Type 1, type 2, type 3, type 4, type 5, right? So why only type 2 is used? Because there are certain regions. Okay, so type 2 is important. Let me write here type 2. Why type 2 are used? Now let me tell you the region. Type 2 are used because the very first one, no ATP is needed for the cleaving action. First of all, this type 2 is no ATP is required. Second thing, that is it make a cleaving or cut in both the strand of DNA molecule. Means when we cut, you can see here, this is the DNA molecule double stranded. In this diagram, just focus, and okay, this is DNA double strand, but it is going to make a cut on both the strand, which is impossible in type 1, type 3, type 4 and type 5. But type 2 have such capability who cut the DNA duplex from both the end, and that's why this DNA is only restriction enzyme type 2 are used in the gene manipulation. Here, so I can write another. It make cut on both the strand. This is the only 2 region that type 2 are used in a restrictions into nucleus for cutting purpose. Clear? Okay. So this is type 1, type 2 and type 3. Total 3 main type as written over here, but now it was total 5 in number, so you can just make a correction slightly. Total 5 main type of restrictions into nucleases are present nowadays. So I'm just writing here type 4 and type 5. Okay, total 5. Okay. Now let's understand little bit important characteristics of all 5 type of restrictions endonuclease. Now type 2 is very much clear for you, right? Now come and discuss the type 1, type 3, type 4 and type 5. When I say type 1 is enzymes cleave at a random site for away from the restriction site. Like suppose in bacterial DNA there is a restriction site, right? So type 2 is not going to cut at the restriction site. It's going to cut slightly away from the restriction site. Suppose this is a restriction site, so it is going to cut away from the restriction site. Okay, and that's why they are not used in the recombinant DNA technology. So this type 2 is never used in recombinant rDNA technology. Is this clear to all of you? Okay, come to the type 3. Now when I say type 3, these enzymes also cleave at a site short distance from the recognition site and far away from the restriction site. Means if you want to cut, see, this is a DNA, bacterial DNA, and your desired, this point, this is your desired location. Okay, just imagine this is the desired locations. But this type 2 is not going to cut your desired location. It is going to cut slightly away from your desired location, and that's why this is also not suitable. Clear? So you have to remember that type 3 enzymes all cleave at a site a short distance away from the recognition site and far away from the restriction site. Okay, that means of it is this one, type 1 is slightly away. This is slightly away from the restriction site, but type 2 is far away from the restriction site, from the restriction sites, and that's why this is also not in use in a rDNA technology. Now come to the type 4. When I say type 4, these enzymes cleave only methylated DNA. This enzyme type 4 is always cleave at methylated DNA. Now what is methylated DNA? That means of DNA band having methyl group at their end. That is what methyl cap, we can say cap 7. You might have heard about the cap 7, right? So cap 7 is methylated DNA. So if there is bacterial DNA and that is methylated DNA is present, then only this type 4 is going to work, otherwise it is not working. So it is only cut at the methylated DNA, and that's why it is also not used in rDNA technology. Clear? Come to the next and the last type, that is called type 5. Now type 5, these enzymes basically use guide RNA to target the specific non-palindromic sequence present on the invading organisms. Like here you can see type 2 is always cut at the palindromic sequence, right? As you can see, this is the palindromic sequence, yes or no, right? But type 3 is not going to cut, it is always going to cut non-palindromic sequence. Okay, and that's why it is very specific in their use. Okay, so these are few major tools that is used in restrictions into nucleus as a molecular features, and out of this 5 types, 2 is very important and rest all are like there is a very less amount of use of that organism which is never used in a RDNA technology. Hope this is clear about the enzymes used in RDNA. Yes. Okay. Now let's talk about the nomenclature of restrictions enzymes. Nomenclature of restriction enzymes, especially type 2. Type 2 is restrictions enzymes are named for their bacterium from which they have been isolated. So the first letter you can see, type 2 restrictions endonuclease enzymes, right? Type 2 is called eco R1. Now what is mean of this eco RI? So I means it is a Roman word, numerical word. So eco that stand for Escherichia coli, right? So the first letter used for the enzymes for the that is for bacterium genus species, right? First letter used for the enzymes is the first letter of bacterium genus name. What is the bacterium genus name? Escherichia, right? So Escherichia, the first letter is E. Escherichia coli. That means of Escherichia coli is a bacteria. E. coli is a bacteria through which this restrictions enzymes is obtained, right? And that's why the name is capital E, stand for Escherichia coli. Then come the first letter, that is 2 letters are to its species name. Now what is species name? As I told you that Escherichia coli. Shortly it is called E. coli. So E is a genus and coli is a species, right? So first letter is E, then CO stand for coli. So coli, initial 2 letter from the species, and then R. The fourth letter of the name enzyme is first letter of the strain. That means of what kind of strains are used, and it is always written in the capital. So R stand for strain. Okay, R stand for strain, that which strains are used, whether it is a first strain, second strain or third strain. And here you can see first if I is written, that means of it is a Roman number, the order in which the enzymes were isolated from the strain of bacteria. Okay, so if I write eco R second, what does it mean? It means it is the name of the bacteria is Escherichia, species is coli, strain is 2. Are you getting my point? This is how the nomenclature of restrictions enzymes made. Okay, similarly you can see okay, another you can see enzyme eco R1 was isolated from the bacterium Escherichia coli, RY13. Enzyme eco R1 is named similarly, right? So this is how the simple nomenclature. There are certain enzymes also used to nomenclature, but these are the common method to nomenclature any particular enzymes. Okay, any doubt in this? Come on, respond it, yes, no. Okay. Another you can see the next enzyme is, okay, this is not a Hindi 2, it is a Hind 2. Hind 2, it is not Hindi 2. It is written as Hindi, right? Yes. So you can see here the first excision endonuclease is Hind 2, and it cut DNA by recognizing a specific sequence of 6 base pair. This is called the recognition sequence for Hind 2, and you can see the resistance site and their particular regions, or we can say the resistance site is 5-GTPU PYPU AC3. This is the sequence of Hind 2. There to remember. Though it is not important, but you can remember. Now this is what is palindromic sequence. The restriction endonuclease recognizes specific palindromic sequence, nucleotide sequence, and it is sequence of base pair that read the same on the two strands. So you can see when you read, how you will recognize the palindromic sequence. When you start reading from 5' end to 3' end, what is, how you can read 5-GAATTC 3', and when you read from this side it is also same. See 5-GAATTC, right? So this is the way to recognize the palindromic sequence. Always keep in mind. Okay, palindromic sequence is, it is the sequence when you read from the both side it is same. Oh my god. See, Sasidharan mentioned in the chat box that, sir, can we have a break till 8:35? Now he instructed me that, sir, please give a break till 8:35. Such a great.
No, sir, not like that.
Yes, Sasidharan?
No, not like instructing us also.
Okay. So, alright, I also decided that yes, now I will give you break, not for 35, but I will give you a break for at least, now it's a 29. Okay, okay. No problem. You can take a break till 8:35, and then we'll resume, and then we'll see the remaining concept of today's. Okay. So you all can take a break. Anthony, so silent. Anthony, are you there? Rest all can take a break. Yes. What happened, Anthony? Anthony. Hello. Hello everyone. Now break is over. Yes, all of you please on your front camera. Hello. 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 Am I audible to all of you?
Hello, students.
Hello.
Hello. Hello. Please give response to sir. Hello. And Sasidaran Sarjana.
I
Hello.
think so.
I'm audible to you.
Most of them are in the lobby. Anthony, then Sindhuja Sri and Gopinath all in lobby. Only Sarjana and Sasidharan is. Hello.
Yes, sir. They are not given access to the camera and mic. That's why they are in lobby, sir.
Okay. But they even not responding. Hello.
Hello, students. Give response to sir. Sir is asking you. Hello.
I got response from Sindhuja. I'm here. Okay. So what about Anne? Anne, are you there? Okay, Anthony responded good. Okay. Okay. You all are in the lobby, I think so. But anyways, you can hear me. So let's continue. So as I told you about the restrictions into nuclease enzymes, and you can see restrictions into nuclease enzymes cut the strand a little away from the center of palindrome site. Like this is your restrictions into nucleus with the help of restrictions into nucleus. Now why I need to show the SAGEs over here, because SAGEs stand for it's a restrictions into nuclease enzymes. It is a kind of enzymes who working on the DNA to cut the DNA at the specific site, right? So it cut slightly away from the palindrome site, but the between the same two bases on the opposite strand. This leaves the single stranded overhanging stretch at the one end, and because of this you can see it is the sticky end. So this is called the sticky end. You can see this portion of DNA is not there, only the lower portion DNA is there. So this is sticky end for this DNA segment, and this is the sticky end for this DNA segment, right? And these two sticky end can join with another segment of DNA, so they are called the sticky end. And this sticky end form H bond. This is also important. H bond. Sticky end form H bond with their complementary cut counterpart, and this stickness facilitates the action of DNA ligase. That means, so with the help of now these two DNA is not going to join by their just matching. They need a certain glue to stick, and that glue in the form of DNA ligase who going to stick this two strand of DNA as a recombinant DNA. So always keep in mind DNA ligase is a sticky, it's kind of glue who work as a sticking of two different parts of DNA. So when cut the DNA, the same enzyme restrictions enzymes, the DNA fragment have the same kind of sticky end, and they are joined by the DNA ligase. So as I told you that DNA ligation is very important, and that's what the name is ligase. That process is called the ligations. and the enzymes uses DNA ligase. So you can see this is the DNA backbone. This is taking end of DNA insert. So these two DNA you can join together with the help of ligations, and these ligations become your recombinant DNA segment. So you require DNA ligase. Okay. Now after this we need to understand one important concept, and that is called the gel electrophoresis. Now gel electrophoresis, or we can say the separations and isolation of DNA fragment. Now why we need to use this gel electrophoresis in biotechnology or in RDNA technology? Why we need to use the gel electrophoresis? Now you know that DNA restrictions endonuclease is a kind of enzymes who going to cut the DNA at different segment. Just pay attention, Sasidharan. Don't watch TV. I know the TV is in the background. My God. So why this gel electrophoresis is important? Because see, this is the isolated DNA, right? With the help of enzymes, restrictions endonuclease enzymes, this DNA is going to cut into a fragment. Now, but I don't know that what is the size of this fragment, right? I don't know the size of the fragment, because size is going to decide, and that size is going to insert into the vector DNA, right? So you need to understand that what is the size of this fragmented DNA. So how you will, is it possible for you to measure the DNA by scale? No, it is impossible to measure by the scale. So therefore you need a gel electrophoresis to separate the DNA according to their size. Okay, so what is gel electrophoresis? It is the process of separation of DNA, RNA or protein fragment under the presence of electric field is known as gel electrophoresis. Clear? That means of this gel electrophoresis in a liquid form dissolve in a certain ethanol or methanol, right? So it is impossible for you to separate the DNA from that according to the size. So what you have to do, you have to just with the help of micropipette, you can load your desired DNA on this gel. Okay, this is your gel, so you just load it over here. What will happen when you supply the electric current in this gel electrophoresis? This band is going to separate according to their molecular weight, according to their size. So those who are smaller in size, they move more faster towards the positive, towards the cathode to the anode, right? So it's going to separate according to their size, and once it's separated, you just remove the gel and you can extract the DNA accordingly. Clear? So this is a technique of separation of DNA fragment according to their size. So let's learn more about it. So gel electrophoresis, the DNA segment is need to be separated into the different fragment according to their size. Why you are using the gel electrophoresis? You are going to use the gel electrophoresis to separate the DNA according to their size. This is very important. And then the desired fragment need to be separated. Like here you can see, this is the gel and this is the loading well where you have to load the DNA sample, and then when you start the electric current, it will run according to the size on the gel, and you can see those who have low molecular weight, they run faster. Those who have high molecular weight, they run little slower. So this is what a kind of fragment that you can separate according to their size. Now, what are the process of this entire system? What need to be prepared? So very first, as I told you, that it having a two important position. One, that is positive charge and negative charge, right? You can see in the DNA also, the positive, and this is the negative charge, and this is the positive charge, right? So there is a positive and negative charge is there. Now, what is this positive and negative charge? Positively charged molecule move towards the cathode, and negatively charged molecule move towards the anodes, right? So it is passing through the matrix or media. Now, what are the matrix and media? That media and matrix is in the form of gel, and that gel is made up of from agarose. Agarose, you know agarose. What is agarose? Agarose is a polysaccharides matrix which is obtained from seaweed. So agarose is a kind of, it is a powder form which is extracted from the seaweed, and this agarose is used for making a gel. Even agarose is sometimes also used in various confectionery candies making industries, right? So this agarose is used in making this gel. Clear? Okay. Now here you can see what are the materials required to perform the gel electrophoresis. So first things is you require a desired DNA fragment to be cut. Means suppose any DNA which you want to separate according to their size, you need a fragment of the DNA. Then you need a agarose gel, you need a gel plate, and ethidium bromide. These are the important materials which is required. Okay, even you can see in the picture that this is a typical method of digesting the gel, and then you can make a agarose plate. Now how you will make this agarose plate? You can see there is gel agar casting instrument is available, which is in the laboratory. So you can just pour this liquid into that, and you solidify it, become solidified because it is a semi solid, so it becomes solidified, and then you make a well inside this. So you can see this is the well where you can load your desired DNA, and then you can run with the electric current, and then it will start running, and it will separate it according to their size. Now, what is the principle involved in this gel electrophoresis? So DNA being negatively charged and move toward the positive electrons. So anode under the influence of electric field. So you can see here, this is the complete set of electrophoresis. What is this complete set? This is your gel plate. This is a gel plate. Now you have your DNA into this vial. Okay. Now with the help of micropipette, you can just load this sample into this well. So here is well, here is another well, here is another well. You just load it into this gel, and then you supply the electric current, cathode and anode. So you can see here, those who are shorter molecules, or we can say low molecular weight, they run faster and move towards the anode. Always keep in mind that gel electrophoresis, DNA is always run from cathode to anode. Cathode to anode. You know why? What is the reason that it run to cathode to anode? Tell me. Why it run cathode to anode? Anyone know? Very good. And DNA is a negatively charged. So if DNA is a negatively charged, then it runs towards the positive charge, and that's why it is running to the anode. Please, all of you remember, DNA is a negatively charged, and that's why you can see DNA is loaded at the negative region. That's why DNA is loaded over here, not at the cathode side. And when the electric current is supplied, you can see the band size. This is all the band size. Can you see here? This is all the DNA band. It loaded according to their size. Like here. This is all the DNA band which is separated according to their size. Are you getting my point? Yes. Okay. So this is what actually principle involved. It is based on the negatively charged, because the DNA being a negatively charged move towards the positive electrodes and the anode under the influence of electric current. This is the principle behind this gel electrophoresis. Now, what is the procedure? How it works? So, see, the process is also very simple, as I've already mentioned many times, the simple procedure of DNA, but let me tell you again. So first of all, well, DNA restrictions enzyme and ethidium bromide is required for this work. Now you have to just cut your DNA. Okay, let me write the procedure for you. Yes. These are total 6 steps of DNA procedures. What is the 6 steps? Let's see here. First of all, you need to cut the DNA. Okay, total 6 steps, total 5 steps are there. First is you need to cut the DNA fragment separate under the influence of electric field. Then the negatively charged DNA molecule move towards the anode. You know what is the region. The large DNA fragment are present near the well, and the smaller DNA fragment that move towards the away from the well. So what is well? Present near the well means that is the loading type where there is a negative charge, and it move away from the well, that means of it reach to the positive charge. So those DNA present at the anode side, they having high molecular weight, and those who present at the positive side, or we can say at anode, they are having the low molecular size. Clear? Now the orange color band of DNA can be seen when exposed to UV light, and then the separate strand of DNA are cut into a agarose gel and extracted from the gel. This is known as elution. This is known as a elution. Clear? Like here. So this is the DNA band, cathode, anode and all. This is the complete setup. Orange color because it is a dye, and when you observed under the UV light, okay, it look like it is under the UV light, so it, because the inflorescence, it look like as a orange. See here, the separate DNA fragment can be seen only after staining the DNA with the compound known as a ethidium bromide. You cannot see the DNA band with your naked eye. So to visualize the DNA, you have to stain that gel with the ethidium bromide. And then this ethidium bromide, when you keep inside the UV light, UV radiations, you will get bright orange color band. Is this clear? And good. Now, here you can see this is cathode, this is your loading well, where you loaded your DNA fragment, and this is your anode where the DNA is run according to size. So those having the low molecular weight, it is here. All the low molecular weight DNA is present over here. This is the medium molecular size, and this is the maximum. So they are going to separate according to their size, right? So the separated strand of DNA are then cut. Now this is the separate strand. Now you have to cut it. So cut it from the agar gel and extract it from the gel piece. And this process, when you extract the DNA from this gel piece, this process is called the elution. Elution means removal of absorbent. So don't forget what is elution. Elution is removal of absorbent. Is this clear to all of you? Yes? This is all about the today's topic. Now, if you have any doubt, then please ask me your doubt. Anyone? Any doubt? Yes? Gopinath and Sindhuja, Anthony, Sasidharan. Anthony, why you joined late session? Anthony, why you joined late today? Not feeling well. Okay. So today I'm also not feeling well, but I joined on time, you know.
Can you hear me? Sir,
Yes.
sir, I was I got typhoid hospital, sir.
Okay. Okay, typhoid. So you can just say me that you are suffering from some kind of disease, then okay. Otherwise, see, I can say also I'm not feeling well. Okay, then fine. If you are seriously like, so you can just mention, no?
Yes,
Okay, all right. So now you're feeling good?
for medication, sir.
Okay, okay, good, good. Take proper medicine.
Yes, sir.
Oh, good. Now, so anyone have doubt? And said no doubt. I want response from all of you. If you have any kind of doubt. So what we completed today, we started with the a simple history behind the biotechnology. We also studied the principle of biotechnology. We also studied that what are the basic aspects of biotechnology. They are basically work on two important principle concept. One is genetic engineering and their bioprocessing. Right. We also studied about the basic steps in a RDNA technology. So what are the basic steps? Total 3 steps of RDNA technology we studied. Right. Followed by we studied that the tools used for biotechnologies. So there are basically 3 tools are used. One of them is enzymes, which we studied in details about that enzymes, like exonuclease, endonuclease, then restrictions endonuclease enzymes. Right. We also discussed about the gel electrophoresis, started from their like materials required, what is gel electrophoresis, what kind of material required for this gel electrophoresis, what is and what is the constructions and how it work, the working principle, how it work, all we have studied about the gel electrophoresis and why we need to use this gel electrophoresis in RDT technology that we studied. Yes or no? In the next session, we will learn the various kind of cloning vectors, especially PBR uh, 322 and all that we will learn in the next session. But there are maybe a question based on the PBR 322, which is a kind of vectors in today's session also. So if there are such questions, then please let me know. So I will try to give that questions again in the next session also, right? So in the today's class test, maybe that question is there. One question is there that I will. You can just tell me the number of that questions. I will rectify that. Okay. So this is all about the today's session, that is biotechnology principle and processes. Now let's try few questions in the form of assisted practice. Now the question is given to you: which one of the following represent a palindromic sequence in the DNA? Now here you have to identify the palindromic sequence, and I have told you how to identify the palindromic sequence. Yes or no? And I want answer from Kirtini. Kirtini, please respond it. What is the answer of this question? Hope the question is visible to all of you, right? Sindhu just okay. Sasidharan, what is the answer? You can respond in the chat box. Okay. How you recognize that A is the right answer? Tell me. Yes. Okay. Kirtini not responded. Kirtini, please respond it. Kirtini and Gopinath. Okay. So most of you responded A, and that is absolutely right, because palindromic sequence is those sequence which you can read forward and backward. It is same, like 5 dash and GAATTC 3 dash. This is one way. When I read from this side, 5 dash GAATTC 3 dash. So this is quite matching. If I read the another one, 5 dash CCAATG 3 dash and 5 dash CCT. Now here it is not instead of A, it is given as T, and that's why this is not the right sequence. Similarly, you can also compare the another one, similarly CATTAG. So CAA, now again 2A is given over here, and that's why this is also not right one. Then here GATCACC, and when I read from this side, GA, again here A is given instead of T. That's why this is also not the right answer. So the right answer is A. Is this clear how to recognize this? Okay. Go for the next one. Next question. Sasidharan, pay attention on this question. Next question. DNA fragment generated by the restrictions into nuclease in the chemical reaction can be separated by centrifugation, polymerase chain reactions, electrophoresis, or restrictions mapping. Now again the question is very simple, but response is very late. I don't know why. because the question is very simple, so your response should be quick. As Anne responded, and Sasidharan, Sarjana, 3 responded. I'm waiting for others to respond it. Kirtini, please respond it. Yes, all of you please respond it fast. DNA fragment generated by the restrictions endonuclease in the chemical reaction can be separated by, many times I told you that to separate the DNA according to the size, we need or run gel electrophoresis. So yes, C is the absolutely right answer, because centrifugation is not the right, because in the centrifugation we generally use to separate the cell organelle. Centrifugation is used for to separate the cell organelle. Okay, it is basically used to separate the cell organelle. Polymerase chain reaction is used to make a multiple copy of DNA, n number of DNA. Restrictions mapping is used in the genetics, right? So gel electrophoresis is the right answer. Another one, which of the following is a restrictions endonuclease? Which of the following is restrictions endonuclease? Okay, so and Sasidharan, Anthony has responded. So yes, so we studied total two example of restrictions endonuclease enzymes. One is eco R1 and another one is HIND2. This is the restrictions endonuclease enzymes. So this is D is the right answer. This all is not a restrictions endonuclease enzymes. So D is the right answer. Now this is what this should not be reflection. This is your summary, actually summary. What we studied, that is the principle of biotechnology, which includes the genetic engineering and bioprocessing. We also studied about the process of recombinant DNA technology, which includes how the DNA with the desired characteristics are isolated from the organisms, and this desired DNA is incorporated with the DNA of the vectors, which is done as a DNA fragment need to be replicate, and this desired DNA is linked with the origin of replication to the vector and thus multiply itself in the host, so the number is increased, and the newly formed DNA in the recombinant DNA, and this DNA is inserted into the organisms to incorporate the desired characteristics, or we can say the vectors is used for production of genetically modified product. So any product you can develop from this technique, which is RDNA technology, of your desire, of your interest gene. Clear? So this is all about the today's sessions.